MRC Reagents and Services have an important role. We help many scientists in Dundee and all around the world to carry out important research by providing them with proteins and other reagents they need to perform their experiments. This enables them to focus on answering questions like how do proteins work? And what do we do when proteins go wrong? We use bacteria to make proteins that our scientists are interested in studying, and we grow lots of them. We start by putting in the piece of code, the DNA that tells the bacteria to make the protein, and some other features like affinity tags into the bacteria. We do this by using expression vectors, which are like taxis, that drive the DNA into the bacteria. We then grow the bacteria in large flasks that contain media, liquid food, for around 20 hours. A spectrophotometer is used to measure the amount of bacteria by looking at the optical density of the liquid, how cloudy it is. If there are enough bacteria in the liquid, a special chemical is used to send a signal to the bacteria to make lots of protein, and we leave the bacteria for around 20 hours. Collecting the protein from the bacteria takes several steps. First, we remove the bacteria from the liquid they have been growing in by gently spinning in a centrifuge. Then, we need to break open the bacteria cells to release our protein. We add in lysis buffer and use a sonicator, a machine that uses high-pitched sounds, creating vibrations. Together, these break open the bacteria and release our protein. This liquid is spun in a centrifuge at a high speed to separate the broken parts of the bacteria from our protein, which remains in the liquid part. We now need to remove the protein from the liquid, and we do this by using a resin that binds to the tag which is attached to our protein to fish the protein out of the liquid. The rest of the liquid part is washed away because our protein is safely stuck to our resin. Now we need to remove our protein from the resin using a special solution called a Luchan buffer. This contains chemicals that stop the protein tag from sticking to the resin, and we can collect the protein as it comes off the resin. The eluted protein is then dialyzed, a process using a special membrane to remove unwanted molecules from our sample and gets it into the correct solution we need for storing it. We now need to check that everything is okay with our protein. Firstly, we check the amount of protein made using a special dye. The brighter the blue colour, the more protein we have made. Secondly, we check that the size of the protein is correct, its molecular mass. And thirdly, we check how pure the protein is. We run the protein on a special gel to check both the size and purity. If the protein passes these checks, it is then sent to scientists who can now begin their exciting experiments.